Welcome to our second night of Biotech 201 for those of you here in the audience and those of you joining us online as well. For those of you joining us online, I hope you have some really delicious desserts in front of you because here at the Jackson Center, we're doing double dessert duty. We are feasting, I probably shouldn't tell you any of this, those of you online. We are feasting on Italian cream cake, and let me just tell you, the Italian cream cake is from, there was a wonderful celebration of the life and legacy of Lonnie McMillan yesterday here in the Jackson Center. Many of you know Lonnie's the co-founder of HudsonAlpha, and Lonnie had a special fondness for Italian cream cake, and we had some Italian cream cake left, and so the family said, well, let's share it with Biotech 201. So we have pre-break dessert, and then we'll go straight into cookies at the break. And I probably shouldn't tell you this, but tonight we're going to talk about the impact of your microbiome and obesity and diet. But I also knew that if I gave you carrots and celery, that this audience would completely revolt. Again, tonight we're going to talk specifically about our gut microbiome, and we're going to talk about obesity, diabetes, and inflammatory disorders. And then next week, we'll talk about autoimmune disorders and about the relationship between our gut and our brain and behavior. That's going to be a really interesting week. And then our final week, we'll follow up with antibiotics, prebiotics, and probiotics and try to give a little bit of a what is the difference, what do they mean, should we be thinking about taking them, what kind of data should we be looking at. So we're going to actually start tonight with Q&A because I gave you no Q&A last week. And I'm going to start with two questions that were asked from our online audience last week. And the first question was a question about antibiotic usage. And if someone is sick for a long period of time, should they take the same round of the same type of antibiotics? Or should they actually switch and do one antibiotic to start and one antibiotic to end? So that clearly is a question that should start with consult your doctor, not this doctor. I know that there's a lot of discussion about broad spectrum antibiotics that actually kill lots of different types of bacteria versus narrow spectrum that have specific classes that they completely leave alone. I'm going to dig into the research on that so that when we hit week 4, I can give the questioner a better answer. So we're going to put that question on hold. The second question that we got was asking about how rapidly your gut microbiome composition changes when you shift your diet, when you go from being heavy meat eater to, say, vegetarian. And the answer is that based on some of the research that's out there, it actually can shift within a matter of 2 or 3 days. So you've got this population of lots of different kinds of bacteria, some of them just barely hanging on in really low numbers, some of them in huge numbers because you've got all the nutrients coming through your digestive system. When you suddenly shift up and move from, say, lots of animal protein and fat to lots of plant-based fiber, the ones that have been living on the animal protein and fat, they're going to die back and you're going to have really small numbers. They don't disappear, but you're going to have really small numbers. And those small numbers that haven't had a whole lot of plant fiber, because, you know, periodically you might throw a stalk of broccoli in there, now suddenly are going to grow in numbers. And that can happen literally in a 2 or 3 day window. So your gut microbiome is incredibly responsive to shifts in your diet. So let's now take some questions from this audience and questions from our folks online.
Are you going to discuss phage therapy?
Am I going to discuss phage therapy? Probably not, but if I've got space as we hit week 4, it's something that I'm going to try to see if I can slide in. You're welcome. Next question. Really? I have never known this audience to be at a loss for questions.
When you take an antibiotic, does it kill everything in your system?
When you take an antibiotic, does it just like wipe out? Does it like annihilate everything? No, it doesn't. It does knock out large swaths of different types of bacteria, but it is not that you suddenly go from a thriving metropolis to like the Wild Wild West tumbleweed desert. I'll show you some data that shows what happens to the diversity of your microbiome and how rapidly your microbiome recovers, and if it fully recovers, after an antibiotic treatment. Yes? Oh, that's a great question. So we talked about this practice called vaginal seeding, which is for, it's becoming somewhat popular for infants that are born by cesarean section, and you're actually swabbing the infant's skin and mouth with a gauze that has been seeded with microbes from the mother's vaginal canal. And the question that I brought up was that there's been some caution about that because there's a concern that you actually could be giving the infant harmful bacteria if you aren't doing the check for that. And your point is, which is a great one, is that same thing could happen in a natural birth. You don't know that. So it's something that you've just got to be aware of. I think the issue is that right now, until we have a good sense of specifically what bacteria are the beneficial ones, that just randomly saying, hey, let's do this practice is a little bit similar to people saying, when we get to week 4, we're going to talk about fecal microbiome transplants, and people just saying, hey, I'm going to do a fecal microbiome transplant on my own without necessarily being sure that the donor actually doesn't have something that you don't want. So it's that, hopefully that answers that question. Other questions? Yes, sir. Ooh. If I cut myself, sometimes I use an alcohol swab to kill germs. If I take a shot of whiskey, what does that do to my intestinal microbiome? I'm going to need to get back to you on that. But my thinking is that much of the ethanol is absorbed by your small intestine. So by the time it gets to the part of your digestive system where you've got the most bacteria, it's probably long gone into your bloodstream and is no longer actually passing through. But let me dig into that in a little more detail. That does not mean that next week we're going to have shots on the back table. Yes? Would you say that again? Are lectins toxic to the microbiome? I don't know, but my group that is monitoring the online chat, I'm going to ask them to write that down so that I can dig a little bit deeper into that. That's a good question that I would be totally making something up. I know slightly less about that than I do about alcohol in your gut, so we'll just stop right there. One more, yes? If you have a colonoscopy, does it wipe out your microbiome? So as we're going to talk about in just a second, the largest concentration of bacteria in your body are in your large intestine, in your colon. And when you have a colonoscopy prep, your goal is to completely clean out that colon. And part of that also means clearing out a lot of the layer of mucus that lines the villi in your colon. And that's where lots of those bacteria live. You don't wipe it out completely, but you drastically, for a brief window of time, drop the amount of bacteria that you've actually got living in your colon. So if you've ever had a colonoscopy, and then let's say that your first meal consisted of, Brussels sprouts, broccoli, and asparagus, that's a whole lot of fiber passing through your system that you potentially are no longer able to fully break down, and you may have some gastric issues along those lines. But it isn't something, colonoscopy doesn't long-term change the population of microbes that live in your gut, at least not from the data that I've seen. Great question. Not a reason to skip a colonoscopy. You can't go down that road. And I've got one online. What do steroids do to your gut? That's a really good question. I'm going to look into that as well. And I also have one more that says, there's a white Nissan Altima in the parking lot that has their lights on, car tag XDEGA, X-D-E-G-A. So, white Nissan Altima in the parking lot. We will all look away if you need to get up and go turn your lights on. All right, so let's move on. We'll stop and we'll take questions further throughout tonight as well. All right, as a reminder, we talked about that your microbiome is composed of certainly bacteria, but lots of other microbes, not just bacteria. And I've got those on the slide behind me, including viruses and fungi and phages, which is going to come in if we end up talking about phage therapy, how that actually works. Phages are tiny viruses that specifically kill bacteria. They are incredibly numerous. There are probably more bacteriophages in your body than bacteria. So we'll dig into that a little bit more. But just as a reminder, we're going to spend most of our time talking about bacteria, but there's a whole lot more going on in your body besides just bacteria, much of which we haven't fully uncovered. I'll also remind you about our pathway that takes us from, I see a difference between individuals that have this disorder or this trait and individuals that don't, and all the things that have to happen. We have to replicate that in multiple populations and then figure out if there's a functional storyline behind that. Is there a biological reason for that, or is this just part of chance? If I want to think about this in terms of therapeutic approaches, if I start making changes in a model system, like a mouse that traditionally was born with no microbiome, does that seem to give me some sort of difference that I then could take into patient clinical trials and then all the way up to therapy? And as a reminder, most of what we're talking about still hangs out down here. And as we move through each night and each example, I'll give you a sense of where we are in that spectrum. But again, just a reminder, just because you see a paper that comes out down here does not mean that you are ready to take this home and try it in your kitchen. Here is an important challenge, a limitation of talking about the gut microbes. Most of the research around your microbiome is specifically dealing with your gut microbes. And I want to be careful here. Most of these studies are in that bottom box. They're association studies. We have very little insight at this level as to what the actual functional reason why you might see these differences. That also means many times, we don't know if the altered microbiome is a cause of the disorder or if it is an effect of the symptoms that come along with the disorder. So is it chicken or is it egg? We're going to talk about a word called dysbiosis right here. Dysbiosis is an altered set of bacteria, you're out of equilibrium, the bacteria that are in your gut. You have more of something that you wouldn't traditionally have, or you're missing things that you normally ought to have. So this concept of dysbiosis really forms the central tenet of people thinking about why your microbiome and disease may be so closely linked. But the question, again, is dysbiosis, this alteration, a cause or a byproduct? The other challenge, and we alluded to this last week, is that there is a lack of standardization around how we even test and what we are testing for, which means it's very, very difficult for scientists to compare the results from one study with the results from another. Even if they're looking at the exact same disease, even if they're looking at the exact same way that they collected the bacteria, even the way that they may analyze the DNA, the filters that they use, they may end up with totally different results simply because they filtered in a different order. So I just want to be careful. We have yet to get a good handle on this is the way that we need to collect a sample under the following conditions, and this is how we always need to analyze it. That is incredibly frustrating to you in the audience. It's frustrating to me. It's frustrating to our scientists until we get a standardized approach, and we're beginning to now move in that direction. Much of this is interesting, but not necessarily something we can take to the bank. Case in point, this paper came out yesterday in Nature magazine, a new genomic blueprint of the human gut microbiota. And this work, this is a story, a discussion about it on a website called Genome Web. Essentially, what a group of scientists did was they went back to 75 previous studies where people had published on the gut microbiome of individuals that either were healthy or that had disease, and they had sequenced all of the DNA across all of the bacteria. So if you remember last week, I showed you that chart that started out over here with we can just culture it, and then we can begin to do a little bit of genetic testing, and then more and more testing and more detail. That's on this end of that spectrum, doing the entire genome of all the bacteria. So they went back to these 75 studies, and they took the raw data and they reanalyzed it. And they said, what doesn't match the known database? Because what these other studies did, they looked at the sequence, and then they compared the sequence to the known database of bacteria and said, do we have a match? And anything that didn't match in the previous studies, that fell out of the data analysis because there wasn't a logical match to a known bacteria. This group of scientists compiled these 75 studies. I think it was almost 12,000 humans that had their gut microbiome sequenced. And they said, okay, what's the stuff that didn't match? Let's dig into that and let's see if we can identify potentially new strains of bacteria that haven't been in the database before. And they identified almost 2,000 new species of bacteria that we didn't know because we couldn't grow them in a petri dish. They weren't culturable, and we hadn't had previous data on it. So 2,000 new strains have now been added to the database, which means many of these studies, and as we start saying, I'm looking at individuals with this disorder and I'm looking at their microbiome, we now have a whole new set of bacteria that we can go back and compare against. So this field is far from settled. This is a paper, again, published yesterday. 2,000 brand new species that we had no awareness of. We have discovered through sequencing the genome 2,000 new species. Many of them are similar to existing bacteria, but they're slightly different. Okay, now let's focus on our gut microbiome. The bacteria in your gut play a significant set of roles. I've got 6 of them on the screen behind me and in your handouts. And we're going to dig through many of these. In fact, I think we're going to dig through all of these over the next 2 weeks. So some of these we're going to hit tonight, and some of them we'll touch on tomorrow night. This is, I realize you cannot read this, and I certainly do not expect you. We're going to have lots of Latin words tonight. Lots of Latin descriptions, the species names of our bacteria. But here's what I want you to notice. As you move through your digestive system from the stomach through the duodenum and the different parts of your small intestine, large intestine, and into your colon, you see these numbers here, 10 to the 4th, 10 to the 3rd, 10 to the 4th, 10 to the 5th, to the 7th, etc. And these are colony-forming units. Now, the reason that's backwards is because this came out of a European journal, and the European journal, the language, this was a French article, colony-forming units is in a totally different order in France, the words. But this is essentially rough estimates of the number of live bacteria that are present in these different parts of your digestive system. And so you can see as you move through your digestive system, the exponents, the 10 to the powers, get bigger. And we got 10 to the 10th to 10 to the 11th live bacteria per gram of gut content. Remember, I told you that your intestines, your intestines are made of lots of these little folds, lots of these little villi, fingers. And if you unfolded all of that, your intestines would stretch across a tennis court. Huge amounts of surface area, all of which are covered with bacteria. Your colon is the bacteria-ist part of your body. And this just gives you a sense of what some of the different types of bacteria are that are prevalent in each of those. Think about your stomach has a much higher pH because of all the stomach acid. So the bacteria that are found in your stomach are bacteria that specifically thrive at high pH. As we move through your intestines, pH begins to drop. The amount of water that's available as your large intestine begins to absorb water, the amount of liquid that's available begins to drop. So you have bacteria that have found niches, places that they really thrive in based on the conditions, the local conditions moving through your intestines. I'm going to tell you a story that I find fascinating. So this is, remember last week I told you about the book by Edie Young that I recommended, which I have just totally blanked upon. Say that again? I Contain Multitudes. Thank you very much. I Contain Multitudes. This story is in his book. But Ed also was a science blogger for Discover magazine and then for National Geographic. You can find his science blogs just by searching Edie Young blog. And this is a specific one that he also told when he was working at Discover. And it's fascinating. It talks about this specific type of bacteria, Zoubrella. And Zoubrella is a bacteria that thrives on seaweed, because Zoubrella contains genes in its genome that allow it to specifically digest a polysaccharide that is found uniquely in seaweed. So Zoubrella has the ability to break down this polysaccharide thanks to these genes. I think they're called porphyrins, these enzymes that break it apart. So it gives it a food system. I'm cutting in and out, aren't I? No, it's just in my head. Okay, well, good. All right. Well, all right, so that's an interesting side note. All right, so Zoubrella lives on seaweed, and it lurks on seaweed. That's even better. It just lurks on seaweed because it can digest seaweed. Now, in Asian diets, seaweed is an important food source. So seaweed is often pounded flat into sheets that are rolled into sushi. And not that long ago, the seaweed was not, what's the word I'm looking for? Pasteurized, sterilized, for lack of a better word. So if I were to eat food made from this seaweed, I would be ingesting the bacteria that live on that food. And that's not that surprising. Everything that you eat, you pull an apple off the tree and you bite into it, you are ingesting some of the bacteria that are living on that apple. So if I were eating this seaweed as part of a native dish, I would be ingesting this bacteria. There's nothing unusual, nothing abnormal about that. Where the story gets interesting is that researchers discovered this specific class of genes that digested this polysaccharide on seaweed, not only in this set of bacteria, but they also found them in a bacteria that lived in the gut, but only found in individuals from Asia, specifically from Japan. So this gene that normally is only found in this bacteria that lives on seaweed, this gene had made its way out of the bacteria on the seaweed and into the gut bacteria of the individuals that ate this seaweed. So that's called horizontal gene transfer. So the gene left the bacteria that lived on the seaweed and took up residence inside the DNA of the bacteria hanging out in the gut. And the benefit of that was that now meant that that bacteria in the gut now gained the ability to digest and get nutrients from the seaweed passing through the diet. So it was a selective advantage. So it probably happens periodically, this horizontal gene transfer, but the only reason that it has become established in the guts of individuals, especially from Japan, is because there was a consistent, continual diet of seaweed that allowed that bacteria to thrive and nourish. And then that bacteria was passed from individual to individual and from generation to generation. You and I, even if we eat sushi a whole lot, don't likely have that gene in that bacteria in our gut, unless we are of Japanese, specifically Japanese descent. Because today's sushi, the seaweed, has been sterilized, and so there's no bacteria living on it. And this transition, this horizontal gene transfer, took place thousands of years ago. So it is this really interesting genetic loan where the human gut microbiome borrowed this recipe from this bacteria that normally lived quite happily on seaweed because it gained a selective value in hanging on to that gene. Bacteria swap genes back and forth all the time. But most of the time, there isn't an advantage for them to hang on to it. There's not a selective advantage in evolutionary pressure for it to be kept and for it to allow it to thrive. But just an amazing kind of story about the relationship between bacteria in nature and the bacteria that live inside our body. We will not be serving seaweed tonight either at the break. All right, so let's talk about your intestine. Let's talk about your the transition as we go from your small intestine to your large intestine. And you can see on the screen behind me a cartoon of that. And we've got these shapes on the left. These are undigested complex proteins and carbohydrates that your small intestine, that your human cells don't contain the enzymes to naturally break down. Your body on its own, based on its own human DNA, cannot break down those complex polysaccharides. Lots of stuff that comes out of like fruits and vegetables and non-digestible fibers. So those pass through your small intestine without being broken down, and they make their way into your large intestine. And there are certainly bacteria in your small intestine, but it's your large intestine where these products really provide nutrients for the bacteria. And in your intestines, you've got this layer of mucus, this mucosal layer. And there actually are, there's a thin layer, and then there's a really thick layer. The thin layer is a little more watery. The thick layer is a little bit more dense. And you've got bacteria that live in the thin layer, and you've got a few bacteria that live in the thick layer. But the thick layer really serves as a barrier to keep bacteria and your cells, your human cells, a little bit more separate from each other. So this mucosal layer is really critical for the health of your intestine. And so you've got bacteria that live in the mucosal layer, and then you've got lots of bacteria that are just hanging out outside the mucosal layer that are kind of moving through your system. And these gut microbiomes are munching on these undigested food products that are coming through from your small intestine. Things that your body otherwise wouldn't be able to utilize. These bacteria have a field day with it. They go to town. It's feast for them. And that set of complex undigested molecules varies depending on what you're eating. So if you're eating lots of plant food, there are lots of plant polysaccharides that are coming through. If you're eating a diet rich in animal protein, there's a lot of animal fat and animal protein that necessarily didn't get fully digested that's making its way through. If you're drinking lots of Metamucil a day, there's that fiber making its way through your system undisturbed through the small intestine, and there it is in your large intestine. And we traditionally think, I'm jumping ahead to week 4 a little bit, but we traditionally think the reason we take fiber is specifically because fiber in bulk helps us move things through the digestive system. So we have movement. Movement. And yes, that is true, but the other important thing that happens is that the bacteria in your gut utilize that fiber. It provides them with a great food source, which then allows them to carry out the other functions that are actually really beneficial to the rest of our body. All right, this is a very busy slide that I'm about to show you, and I realize that most of what's in the small boxes you will not be able to see on the screen. You might not even be able to see on your piece of paper. That's okay. What's in those small boxes are just types of bacteria. And I just want you to get a sense of the breadth of the types of bacteria. But I want to walk you through the process of what happens in your gut when things that your small intestine didn't know what to do with show up in your large intestine. So we're going to start here on the left side of the screen. And on this left side of the screen, we've got resistant starch and fiber, so things like broccoli and bananas, or Metamucil for that matter. We've also got mucin, which is the mucus that's in your body. And then we've got amino acids and proteins that didn't fully get digested. These might be plant proteins, they might be animal proteins. So these macronutrients, or host-derived things like the mucus, are then converted from their form that your body can't use on its own by different types of bacteria. And they're converted from these macronutrients into smaller precursors, monosaccharides, smaller sugar units, and something called pyruvate. And the teachers that are watching online, or the teachers that are in our audience, know that pyruvate is a really critical part of the cycle of nutrient utilization and the respiratory process for how we get energy and how we get ATP. So this is a major energy source for your cells that you would not be able to get were it not for this set of bacteria that were converting these things that your body normally couldn't touch. And then these precursors are converted by lots of classes of different groups of bacteria. They take those small molecules, those precursors, and they turn them into things like acetate, butyrate, propionate, all of which are energy sources, and formate and lactate, which are also used by the body in other ways. So essentially what the bacteria in your large intestine have done is take things that your body doesn't know how to process and turn them into energy. Specifically, we're going to talk about short-chain fatty acids. We're going to abbreviate that SCFAs. And then these, the process of creating these also produces hydrogen and carbon dioxide, and a whole nother set of bacteria actually convert them into things like hydrogen sulfide and acetate and methane, which has to leave your body. Methane is a gas that has to leave your body one way or another, and we're just going to stop that conversation right there. But this is what your gut bacteria are doing every moment of the day. They are busy munching away at the things that normally would just pass unutilized through your body. And they are turning them into things that your cells, not only the cells of your digestive system, but the cells across your body ultimately utilize. It is a beautiful relationship. You, as the host, provide a safe place, a place that's comfortable, that's moist, that's warm. Stop there. That has lots of food, and in return, these bacteria thrive and they provide things back to you as the host. They are giving you out of their abundance. It's almost like we should hold hands and sing, you know. But, okay, short-chain fatty acids, those things that I had in this spot on that previous slide. Short-chain fatty acids produced in the gut. They're taken up by the cells of your intestines. So the bacteria living in and around your intestinal cells produce these short-chain fatty acids. They are absorbed through the mucus into your colon cells. And some of those short-chain fatty acids, especially butyrate is the primary energy source for your colon. Your colon cells live off the energy produced by the bacteria that surrounds them. That's amazing. That is amazing to me. And then other energy forms, other short-chain fatty acids, pass into the bloodstream, and as they pass into the bloodstream, they head to multiple different parts of your body. So in the lungs, these short-chain fatty acids decrease inflammation from allergies. In the heart, kidney, and muscle, they're used as an additional energy source, just like in the colon. They help your immune system recruit and mature immune cells. They also have they suppress inflammation. So these things produced by your gut help reduce inflammation throughout your body. In your liver, they actually provide additional regulatory sources. They help regulate the way that fat is stored in your liver. And in your brain, and we'll dig into this more next week, there are some direct effects and some indirect effects for how these short-chain fatty acids get utilized by your brain, but also how they may modulate your behavior and the way that you feel, and potentially your risk of developing neurodegenerative diseases, which we're going to dig into a lot next week. So, the stuff that you eat that your body can't process, your gut bacteria do process, turn into energy that travels all throughout your body. So again, remember last week we talked about we've got to move away from this concept that all bacteria are bad, and that we must sterilize and get rid of all the bacteria in our body. They are doing us huge favors. Now, the challenge is we can also shift the balance of bacteria that are living in our body, and that benefit of favorable to unfavorable things also begins to shift. And that brings us to this concept of dysbiosis, which I first touched on a little bit ago. So, in a healthy gut, these are the villi, the little protrusions that come out all along your colon. And these fingers are projecting out into the mucus, or sometimes through the mucus, and nutrients are being brought into them. In a healthy body, in a healthy gut, you have a strong collection of different types of microbes. You have a diverse microbiome, and that diverse microbiome does a couple of things. It crowds out space for the stuff you don't want. Kind of like you've got lots of great thick grass, and that thick grass crowds out the weeds. The other thing that it does, you have a thick layer of mucus, and we've already talked about that these bacteria are producing short-chain fatty acids. They're helping train your immune system. They're providing energy across the body. The concept of dysbiosis, of altered homeostasis, out of balance, in individuals with especially certain intestinal diseases, the villi are blunted. They're shorter. The linkages between the cells, and again, your villi separate what's inside your intestine from what's outside in the rest of your body. So it's really important that they are very tightly joined, so we don't have stuff inside your gut leaking out into outside your digestive system. In dysbiosis, you actually have spaces that open up. The junctions, they're called tight junctions, are weakened, and you now actually have the ability for things inside your digestive system, inside your intestine, to leak out into your abdominal cavity. This is called leaky gut syndrome. And leaky gut syndrome is associated with lots of different disorders. You can imagine it is not a good thing when all the things in your gut are now making their way outside the digestive system. In addition, if you throw off the balance of bacteria, you now have lots of space for the things that you don't want to come in and take over. That's especially true when we talk in our fourth week about things like Clostridium difficile, C. difficile, that takes over and is a bad dude. You do not want that living in your gut, but it often takes up residence after you've had an antibiotic, a really strong antibiotic, where you've wiped out many of the other bacteria and you've given it a place to get a toehold. So many of the things that we're going to talk about over the next couple of weeks tie back into this concept of dysbiosis and leaky gut. Now, there are lots of things that can influence that. An infection can cause a temporary leaky gut, although it can become longer term. Lots of use of antibiotics. Your diet, there's a genetic susceptibility to that, your local environment. So I don't want to blame all this on the microbiome, but an altered microbiome is a major cause. And in that, we're actually now moving up in here in terms of the data for that. That's not just associations. There's a fair amount of functional data that shows that altered bacterial communities throw off this balance and create leaky gut, and that in animal studies, giving them certain probiotics or restoring the typical balance tightens up that gut. And there are even some human studies that suggest under certain conditions, the right types of probiotics can take you from leaky gut to tight gut. So we're moving up the chain with some of this data. Other things that your gut microbes do, and then we'll stop in just a couple of minutes and take a break. Your gut microbes produce vitamins, especially vitamin K and several of the B vitamins, things that your own body can't produce. And yes, you certainly can get this from taking multivitamins, but the right bacteria in your gut are producing it for you automatically. So they are giving you nutrients and they're giving you vitamins that you wouldn't be able to produce otherwise. Here's another important piece. Your gut microbes can impact the effectiveness of medicines that you are taking, of the drugs that your doctor is prescribing. And there are different ways that this can happen, and sometimes it's a change that you want to have happen, and sometimes it's a change that you don't. So on the screen behind me, there are multiple different examples of how gut bacteria have been shown to alter different drug involvement. So the first one, for example, many drugs come to us in an inactive prodrug form, and they have to be metabolized or converted into the active form in order to carry out their function. And some of that conversion from prodrug drug to active drug takes place by the bacteria in your gut. They take that drug, they modify it in some way, they may break off a sugar, they may break off some other component, and now it has become active, it's absorbed into your bloodstream, and it carries out its function. On the flip side, other bacteria take a drug that's already been activated and inactivated in some way. And so if the drug is primarily getting through your body, if its primary route of travel and transport to the rest of the body is through the intestine, and you have a set of bacteria that inactivate it, this drug is not going to do you any good. Because it's going to, by the time it travels to your tissues through your bloodstream, it's been inactivated by certain bacteria. There are other drugs that rely on bacteria in your gut to help it absorb through the colon and get into the bloodstream. So its mode of action is dependent on those bacteria getting it into and through those colon cells. There are other drugs that are drug conjugates. They've got additional components attached to it. It's a little bit like going from a prodrug to an active drug, and the bacteria actually break it apart and turn it active. And then there are other bacteria that actually take an active drug like acetaminophen and break it down into something that is actually toxic to your body. And so you may be taking a small number of individuals who take acetaminophen at the prescribed dose end up with acetaminophen toxicity, not because they're taking enormous amounts of acetaminophen, but because they have a specific type of bacteria in their gut that is turning it toxic and accumulating it, and it turns against you. Now, the goal is not to go home and say, I need to take an antibiotic and wipe all these bacteria out so they don't affect my drugs. That is not where I'm telling you to go with this, because you could end up doing quite the opposite. But it's just amazing the things that happen in our gut. And if we can figure out what our composition is in our gut, then that helps our physician at some point soon, hopefully, get a sense of what drug they should or shouldn't be prescribing. All right, we talked about this last week. I'm going to go through it really quickly, and then I'm going to take us for our break. Your gut microbiome has its biggest development when you move from newborn into your first 2 or 3 years. And a lot of that is dependent upon the way that you are born, your method of birth, the way that you are fed, when you're transitioned to solid foods, what that transition is. But by the time you get to about 3 or 4, your gut microbiome looks a lot like your adult microbiome, but isn't fully there. It takes several more years for it to become fully adult microbiome-ish. And then it stays relatively consistent as you're an adult. Remember, I told you that it's very differentiated, so each person in this room has a different set of bacteria that are living in and on their body. They're probably carrying out the same set of functions, but it's very unique. Your gut microbiome, your body microbiome, is distinct to you. Even identical twins do not have the exact same microbiome, as we'll talk about after the break. There's some evidence that accumulates that as we age, we begin to lose some of the diversity of our gut microbiome. and that our gut microbiome becomes more unstable, and there's some really early associations with aging and frailty and an alteration in the gut microbiome. All right, so we're going to take a break. We are going to take a 15-minute break. We're going to come back at 7:30, grab some more coffee, grab a cookie, and we'll start back in 15 minutes. We're going to talk about the impact of diet on your microbiome. So this is an example of you are what you eat, being what you eat impacts what lives inside you, which is really less weird than that sounds. But this is a whole set of studies that have been done, and they actually live probably in this range. The majority of them have actually been replicated. So it's not just I've got one study that I can look at, it's I've got multiple ones that I can look at. And we're going to talk about these 4 diets. There are lots of microbiome studies that have been done. I just saw one a couple months ago about a microbiome diet with looking at paleo diets and those sorts of pieces. But we're going to talk about these 4 kinds of diet. The traditional Western diet, which is lots of carbohydrates, lots of sugars, lots of fats, lots of simple sugars. This is your Lay's potato chips, McDonald's hamburger, and delicious Jackson Center cookies. Our Mediterranean diet, which has a smaller amount of meats and fats and a large amount of fruits and vegetables. A plant protein-based diet, so a vegetarian diet, and then a diet that's very heavy on animal protein. So there is a lot of meat in an animal protein-based diet. So in each of these cases, these are looking at the bacteria in your gut by studying stool samples of the individuals that have been on these diets and then coding them to dietary logs of what people have eaten. Because sometimes we cheat, and you want to get a sense of how faithfully someone sticks to a specific diet. So the first thing that we're going to look at is the amount of bacterial diversity. How many different strains of bacteria are present in the gut from individuals that are on these diets? And diversity increases on the Mediterranean, the plant-based, and the animal protein diet. So there are more strains of bacteria. There are fewer strains of bacteria, less diversity on our traditional Western diet. Now let's talk about specific types of bacteria that seem to be present at higher amounts or lower amounts. Again, lots of words on the screen. We're not going to spend a lot of time on, we're not going to spend any time on specifically what they are, just kind of if you want to dig deeper into them. A lot of work is trying to be done now to get us to this place, to say, okay, well, if I've got more of bifidobacterium, what does bifidobacterium do? What does it eat? What does it produce? What does it secrete that might be in the cells? So what's the relationship between having more or less of those and what that might mean? Another set of data that you can pull from these studies that is perhaps more informative because it is a little bit closer to functional are things like this. So, how much short-chain fatty acids are being produced? On a Western diet, there's a lower amount of short-chain fatty acids, in part because we're eating less plant matter and less fiber. So there's less of that undigestible food making its way to the large intestine to then produce those short-chain fatty acids that have all those other fuels and all those other anti-inflammatory qualities. Higher lipopolysaccharides. Lipopolysaccharides are a protein that's produced on the outside of certain types of bacteria. They've been associated with inflammation and with with damage to cells, higher amounts of inflammation, and a weaker gut barrier, a greater likelihood of leaky gut syndrome. In contrast, there's increased short-chain fatty acid from the Mediterranean diet and the plant protein. The plant protein. There's a reduction in short-chain fatty acids when it's just animal protein. Again, not surprising because much of these short-chain fatty acids come from plant-based fibers, plant-based proteins. Reduced inflammation in the Mediterranean diet, reduced inflammation in the plant protein diet. Inflammation wasn't actually measured in the animal protein studies that I found. The plant protein diet strengthens the gut barrier, so it tightens those junctions between the cells. The animal protein diet, you can see over here, increased TMAO, which is trimethylamine N oxide. This is a mouthful. Trimethylamine oxide is a molecule that is produced as your body breaks down animal proteins. It's been associated with an increased risk of atherosclerosis. And then in red are specific disorders that have been repeatedly associated, replicated with either the Western diet or the animal protein diet. Now, you may have a favorite diet on this page. I just want to make sure that we realize that when this is primarily what our day is made up of, we are doing our gut no favors. This is as close as you're going to get to Dr. Lamb says. And it's pretty close. This long-term diet is not beneficial to your body in a number of ways, but certainly because it does not do you any favors with what thrives in your digestive system. You increase the likelihood of weakened intestinal walls and leaky gut. You increase the amount of inflammation, and inflammation is a driver of so many diseases and disorders, not the least of which is cancer. Ongoing low-level inflammation is a driver of cancer. If you were here when we did our key cancer factors 2 years ago for 201, inflammation is one of those key factors. So I just want to say, if this is what your daily diet looks like, I would encourage you to think about frequently having it look more like this. Throw some broccoli in. Throw some other things, not green beans that you've cooked in lard until they're so soft with a big hunk of bacon in it. Thanksgiving, totally fine. Monday through Friday, perhaps not such a good idea. So the diet, one of the big drivers of what thrives in your gut and then the impact it has on all your other organs is your diet. If you just think of your colon as that place where waste is held in keeping until you head to the bathroom, that is the wrongest assumption that you can make. Your colon is a major, you probably did not think you were going to hear me say, your colon is a major benefit to your body if the right things hang out in it, which is driven by your diet. And I say that fully recognizing I just gave you delicious cookies and Italian cream cake. But I'm making an assumption that you're eating that as a special treat on Tuesday nights. All right. Exercise also increases your diversity of the bacteria in your gut. Daily exercise. There's the key. Not periodic, once a month exercise just to prove that you still got it. Daily exercise increases diversity, increases many of the bacteria that have been associated and replicated with increased health. Higher levels of short-chain fatty acids, lower levels of inflammation, a stronger gut barrier, so a lower likelihood of leaky gut. Here's the challenge, though. All those benefits disappear when you stop exercising. They only hang out while you continue. I don't mean, you can skip a day. And if you get sick, you can skip a few days. But if you don't exercise from, October until February, you are not doing your microbiome a lot of favors. Now, here's the challenge with these studies. As beneficial as they seem to be, most of them didn't control for the other factors that we already know affect your gut microbiome, like your diet, or if you took antibiotics. So you need to take these studies with a grain of salt because there were lots of co-founders. Again, back to that thing I said at the very beginning, we don't have a good level of standardization. Okay, we're going to talk for the next couple of weeks about lots of studies that allow us to move beyond association and further up this box pyramid. And those often involve looking at mice. These are called germ-free mice, mice that were born and have been bred to have no bacteria. They have no microbiome. And then when you give them a specific microbiome, what effect do you get? And what happens when you alter this with antibiotics or with probiotics or with other research studies? So lots of ways that we can begin to get at some of these questions to try to get more to functional validation and intervention answers. So I'm going to spend the next several slides talking about the relationship between your gut microbiome and weight. And we've hit on these if you have been in a recent Biotech 101, because I incorporated this into the Biotech 101 series. This stuff that I'm talking about, we're here and moving further up the box. So you can take mice. This work was first done in 2004. You can take these germ-free mice. They are bred in sterility. All of their food is sterilized. Their water is sterilized. They are born by C-section. Yes, someone is yes, you've got, C-section. Their bedding is sterilized, everything. You can take these germ-free mice and you can give them the microbiome of a typical laboratory mouse. You can do that in a couple of ways. You can do it lavage, so you're actually force feeding it to them. There are some other ways you can do that. One of the least attractive ones is that mice actually eat feces of other mice, so you can just give them some yummy pellets to snack on and they will incorporate anyway. Don't try this at home. But here's what you got. Here's what you saw. So this is looking at the body fat, and in male mice, the fat pads that sit on top of the testes, the epididymis fat pads. And in these images, in each one of these graphs, in white is the amount of fat from the germ-free mice compared to the wild-type mice. So you can see germ-free mice, mice that have no microbiome, have less fat. When you give these mice the microbiome of the wild type, the amount of fat, the amount of body fat goes up. You have not changed what you're feeding them. You have simply given them now a microbiome. And the amazing thing is there's a 57% increase in body fat and there's a 27% decrease in food consumed. Eat less, gain more. Because you've now given them the bacteria that allow them to extract nutrients from things that previously passed through their system undigested. Yeah. For you and I, it's estimated that depending on our microbiome, we get 10 to 20% of our nutrients from our microbiome. So then researchers said, okay, well that's really interesting. There are a set of mice that have genetic changes, genetic mutations, single changes that cause the mice to be obese. So it is a mutant, it's a genetic model of weight gain and obesity. What's their microbiome look like? So they compared the microbiome of these laboratory mice that were wild type, so they hadn't been genetically altered, and these laboratory mice that contained the mutation that caused them to be obese. And when they did that, they saw that the level of two broad categories of bacteria, the Firmicutes and the Bacteroidetes, were different. That the mice that were wild type, that were lean, had a higher ratio of the Firmicutes to the Bacteroidetes, but it wasn't that different. But the mice that had one copy of the mutation or both copies of the mutation, that ratio became way out of balance. Higher amounts of F, lower amounts of B. And that ratio was looked at in lots of different studies, and for a while scientists thought that what they called the FB ratio could be a measure of weight gain and of likelihood of developing obesity. The challenge is that studies in one animal often didn't translate to humans, and we didn't necessarily see the same FB ratio work when we got to a human population. The data were really conflicting, so we stepped back from that some. But again, it doesn't answer the question, is this cause or is this effect? As we dug into the types of bacteria, there was one specific strain of bacteria that kept showing up over and over and over again. Akkermansia muciniphila. So this bacteria, you can tell from its last name, mucinophilia, loves mucus. This bacteria thrives on mucus. It is happy in a heavy mucus setting. And what they identified, let me step out of the way over here, was that it was, remember, this is a log scale. So I am moving up factors of 10 as I move my way up this scale. It was present at much higher concentrations in the gut of the lean mice than in the obese mice. This is the graph all the way on the left for those of you watching from home. And then if we move over to the next graph, this was when they actually fed mice on a high-fat diet. The amount of this bacteria, Akkermansia muciniphilia, dropped drastically as the fat in the diet increased. So there seems to be some sort of relationship between the amount of this bacteria that lives in the gut and the relationship with weight. And when they took mice that had obesity issues and had metabolic disorder and higher amounts of fat and insulin resistance, and gave these mice a probiotic of this bacteria, Akkermansia muciniphilia, what they saw was that the gut barrier improved, so they moved away from leaky gut. It reversed some of the metabolic disorders that were associated with obesity. It reduced the level of inflammation. It reduced the fat mass. And it shifted these mice from being insensitive, resistant to the effects of insulin, to being sensitive to it. So it moved them out of the equivalent of type 2 diabetes, moved them back from type 2 diabetes, just by giving them this bacteria. So now we're here with some of this data. In human studies, everything I've told you about is in mice. In human studies, we actually find some similarities. That if you look at individuals that have been declared clinically obese, and you look at the bacteria that live in their gut, they have increased capacity to extract nutrients. So they are producing more short-chain fatty acids from the food that are passing through their system. There are more Fs and fewer Bs, so that FB ratio, again, not 100% in a correlation, and there are also fewer of these Akkermansia muciniphilia strains. But not every study has shown the exact same results. So this is what keeps us, it's what continually takes us back here before we just jump up to here. Because again, not that level of standardization. Is it controlling for diet? Is it controlling for antibiotic use? Are they using the exact same methods to analyze the exact same set of bacteria? So we think that there are multiple pathways to obesity. Historically, the thinking was you either had to put less nutrients in your body or you had to increase the amount of energy that you burned. It is not that simple of a relationship. There are genetic factors that impact how your body responds to food signals and when it tells you that you have been satiated. There are signals that actually determine how much nutrients you take out. The microbiome seems to play a role in that as well. So there are multiple pathways, some that are independent of your bacteria, some that are dependent on your bacteria, that seem to affect our relationship with weight. We don't have a really good sense yet of what all of those pieces are. So now let's talk about something really fascinating. Let's take mice that are germ-free. They have no microbiome. And let's give them a microbiome, but let's give them a human microbiome. So let's see what happens. Again, lots of caveats around this, but let's see what happens. This particular landmark study took human identical twins, where one pair of the identical twin was clinically obese and the other pair was not, was lean. Genetically, these two individuals are identical, so we can't say that these differences are because of genetics. And then they took the microbiome of each of these twins And they took 2 germ-free mice, and 1 germ-free mouse got the microbiome of the obese twin, and 1 germ-free mouse got the microbiome of the lean twin. Everybody with me on how that study was roughly set up? They didn't change the food they gave the mice. They just changed whether they got a microbiome or not and who they got that microbiome from. The mice who got the lean microbiome had very little change, if any, in weight. The mice that got the microbiome from the obese twin had at least a 10%, on average, a 10% increase in weight gain. The only thing that's different here is which microbiome they got. Now the study gets even more interesting, because I told you that when mice hang out together, part of their socialization structure and their, hey, how are ya, how's things going, is they eat each other's feces. So if you take these 2 mice who have been kept apart and you co-house them, now you have the opportunity for them to switch and take on each other's microbiome. And so they did this. They co-housed the mice for 10 days. And what they found is that the mice who had the obese twins microbiome, who gained weight, when they were co-housed with the mice with the lean microbiome, they could take on the lean microbiome and lose weight, but only if they were fed a diet that was high in fruits and vegetables. If they were fed a high-fat diet, the lean microbiome couldn't win out over the obese microbiome. It didn't seem to go the other direction. So the mice with the lean microbiome didn't take on the obese microbiome. So the lean microbiome was able to resist, and if the diet was right, the obese microbiome was overcome. We're not going to dig through this image because I want to move us through the rest of the story, but that is pretty powerful data suggesting the relationship between diet, microbiome, and weight. Certainly that's not the whole picture, but that is clearly a component of it, at least in mice. We aren't taking humans and shifting their microbiome in this kind of way. Those clinical trials, I don't know if those will ever take place, but that's not on the docket as far as I'm aware of. But some pretty intriguing sets of data from here that would at least suggest if we could shift your microbiome, maybe through probiotics or a fecal microbiome transplant, what might that mean for the amount of nutrients and energy that you would digest, the level of inflammation, leaky gut or not, in a human individual? Let me jump to type 2 diabetes, because I'm running out of time. Type 2 diabetes is very tightly associated with obesity. Obesity rates have tripled across the world in the last 30 years, and type 2 diabetes rates have tracked right alongside them. Type 2 diabetes, often, which used to be called non-juvenile diabetes, your body becomes resistant to the effects of insulin, and you have to have more and more insulin in order to carry out the effects that it carried out earlier. In some cases, you actually have to go on insulin. Not everyone with type 2 diabetes requires insulin, but some individuals do. Many, many cases of type 2 diabetes are associated with weight, and when weight loss happens, often insulin resistance becomes less. And you begin to move back towards being able to be sensitive to the amount of insulin your body is producing. There are a number of studies that have connected type 2 diabetes, individuals with type 2 diabetes, with gut dysbiosis, with altered microbiome profiles. Reduced diversity, different bacteria. Again, in individuals with type 2 diabetes, there seems to be a general reduction in our friend Akkermansia muciniphila. And there's a subset of patients that had higher levels of Akkermansia muciniphila that seem to actually do better in terms of insulin sensitivity. Here's where the story takes an interesting twist. Many individuals that have type 2 diabetes are given the drug metformin. And metformin, so it's an oral glucose-lowering drug. It's known to be effective, but its actual mechanism of action has not been well understood. Relatively recent data actually suggests that it's the bacteria in your gut that are utilizing the metformin drug and actually helping to increase populations of certain microbes in your gut that actually help restore your insulin sensitivity. That doesn't seem to be the only mechanism of action, but your gut microbes seem to be using the metformin or in relationship with the metformin, or the metformin actually allows certain gut microbes, again, our friend Akkermansia muciniphila, to thrive, which seems to allow people to be better responsive to the effects of insulin and to reduce glucose levels. The challenge with that is that if I've got an individual who has a specific disorder and they're taking treatment, their treatment might actually be throwing their microbiome out of whack. In this case, it's beneficial. The metformin is increasing the levels of the Akkermansia muciniphila. But if I'm looking at individuals with type 2 diabetes and saying, is there a difference in their gut microbes, the medicine that they're on could drastically be throwing off what my results are, which was not something that anybody had considered until relatively recently. Gastric bypass is an option for individuals that are morbidly obese. There are multiple different methods. There's one that puts a sleeve around the stomach. The goal is to reduce the amount of, or the capacity of food that can live in the stomach. The Roux-en-Y, I'm probably mispronouncing that, is one of the more common forms of gastric bypass. You can see that up here. You actually surgically take a tiny bit of the stomach along with you and connect it down to the intestine a little bit further down. So you essentially bypass a huge amount of the stomach and part of the small intestine. And this type of gastric bypass surgery seems to be incredibly successful. People lose large amounts of weight, in part because they're taking in tiny amounts of food. But before they begin to lose weight, they begin to have better blood sugar control. Before they begin to lose weight, they often move out of type 2 diabetes. And the data suggests that it has a lot to do with you've altered their microbiome. So think about it. You've taken a whole section of their body and you've totally bypassed it. And now the food that's coming into the large intestine is very different than the food that would have come through had it come through all of the small intestine as well. So you've changed the pH, you've changed the consistency, you've changed the amount of digested and undigested food. And the data suggests, both in humans and in mice, that this kind of surgery, so there are researchers that actually perform gastric bypass surgery in mice. Same kind of people, I guess, that perform C-sections in mice, but that they have found that you're completely shifting the gut microbiome. And if you take a germ-free mouse, a mouse that has no bacteria, and you now give it the microbiome of someone that's gone through gastric bypass, it loses weight. Loses weight. Again, I'm not telling you that you need to go find your friend that's had gastric bypass surgery and ask for some of their microbiome so that you can drop a few pounds. That is not where I'm going with this. But again, there is an accumulating level of evidence that ties your microbiome to the amount of energy you extract and your weight and many of these other factors. And I've already told you the driving thing that impacts your gut microbiome is what you put in your mouth. So this is a really important point. What you eat is a key driver of your health via your microbiome. And we've got to pay better attention to the things that we put in our body. It is not simply because we get energy from it. It has all these other impacts. So our friend Akkermansia muciniphila, lots of studies are now showing that this is really beneficial. And people are beginning to think about, way up there on top of that box, is this a probiotic that we want to start giving to people? How would we give it to people? How would we ensure, is it a specific strain of Akkermansia? Are there other ones that wouldn't be very helpful? How would we ensure that it gets into the gut and is living comfortably in the gut? How do we make all that work? It's also important to note that there was a set of studies that came out that associated increased Akkermansia muciniphila with multiple sclerosis and Parkinson's. When those studies were dug into more deeply, that was not true. That was an association, but it wasn't replicated and there was no functional validation. Again, that's a reason why lots of stuff down here in this box we don't take as the gospel truth. All right, let me wrap us up. Intestinal disorders, things like irritable bowel, inflammatory bowel, and celiac disease, there are lots of associations suggesting that altered gut microbiome is associated. But again, it's associated. We don't have a lot of causative. Do I think we'll get to causative? In some cases, I think we definitely will. We just haven't gotten there yet. But there's an accumulating level of evidence that this dysbiosis, altered gut levels, seem to play a role or may play a role in these intestinal disorders. There are lots of non-bacterial things in your gut as well. We're just starting to scratch the surface of that. And I just want you to know that this information is out there. And some of it seems to be associated with your diet, as you might expect. The type of fungi that live in your gut depends on what kind of food you're providing for them to be sustained on. Next week, we're going to talk about autoimmune disease. And we're going to talk about the relationship between your microbiome and your growing immune system. So before you reach for that antibiotic wipe or that antibacterial hand soap, antibiotic wipe, antibacterial wipe or antibacterial hand soap, especially for kids and grandkids, we're going to want to talk about this because we may be creating a world that is so sterile for our youngest inhabitants that their immune systems are unable to determine what is friend and what is foe and what is self, and it just attacks them all indiscriminately. We're also going to talk about neurodegenerative disorders, and we're going to talk about a relationship between your microbiome and colon cancer that's relatively recent. I'll be up here if you've got questions. I've got a genetic counselor, Veronica, that's going to be up here as well. If you've got specific family health related or medical questions, she's your go-to for that. Have a great week, everybody, and I'll see you next week. Thanks.